Welcome. Today, we're diving into an explainer of a chapter in science that honestly feels like absolute magic. I mean, picture an invisible superhero technology quietly working behind the scenes to feed the world, rescue endangered species, and even mass-produce life-saving medicines. It's called plant tissue culture, and the whole thing starts at the microscopic level.
Okay, let's dive into this. We're going to go on a journey today. First, the superpower of totipotency. Then we'll hit some major milestones in plant culturing, mix up the ultimate plant recipe, walk step by step through tissue culture, and finally, look at some absolutely incredible real-world applications.
Section 1. The superpower of totipotency, a true cellular superpower.
Now, to really appreciate what we're talking about, just think about our own human limitations for a second. If you accidentally lose a finger, you can't just plant that severed finger in a pot of soil and expect it to grow a whole new clone of yourself, right? But plants? Plants possess this mind-blowing regenerative superpower that just totally defies those biological limits. It's called totipotency. It's literally the ability of a vegetative cell to divide, differentiate into any type of specialized cell, and regenerate into a complete whole plant. Seriously, without this one unique cellular ability, the entire field of plant biotechnology simply wouldn't exist. It is the absolute engine driving this whole science.
Section 2. Milestones in plant culturing. Let's look at some historical breakthroughs. Now, unlocking this cellular superpower, that didn't happen overnight. It took decades of scientific trial, error, and honestly, some brilliant deductive reasoning to figure out exactly how to keep these isolated cells alive outside of nature. It all kicks off way back in 1902 when a German botanist named Gottlieb Haberlandt dreamed up this crazy concept of in vitro cell culture. He proposed that plant cells could just keep growing uninterrupted on artificial media. And even though he couldn't fully prove it back then, his predictions were so spot on that he's rightfully known today as the father of plant tissue culture. From there, the dominoes really started falling. We see White formulating the first synthetic medium in 1937, Skoog and Miller cracking the code on how hormones control root and shoot differentiation in 1957, and then finally, Murashige and Skoog developing the incredibly famous MS medium in 1962.
Moving on to Section 3. The ultimate plant recipe. Feeding isolated cells.
You see, you can't just drop a plant cell into a sterile glass jar and cross your fingers. You need to feed it. You need a highly engineered plant baby food, so to speak. Think of nutrient media like a perfectly balanced protein shake that gives those undifferentiated cells the exact fuel they need to survive and grow. The breakdown for that famous 1962 Murashige-Skoog, or MS, medium gives them exactly the building blocks required for life. On one side, we've got the heavy hitters, the macronutrients. These are required in large, millimolar amounts, things like nitrogen, potassium, and a carbon source, which is usually sucrose. Then, on the flip side, we have the micronutrients. These are required in super tiny amounts but are absolutely vital. We're talking elements like iron, zinc, and copper. Mix all of that with some vitamins, amino acids, and a gelling agent like agar to make it solid, and boom, you've got the perfect environment.
Now, what's really interesting about this is the chemical tug-of-war that happens next. It's not just about giving them food, it's about giving them instructions. Scientists actually use plant growth hormones to dictate a plant's morphogenic fate. So if you tweak the recipe to have a high level of auxin, the plant cell gets the signal to start rooting. But reverse that and use a high level of cytokinin, like kinetin, and it triggers shooting, growing the leafy top half. By just adjusting these two opposing hormones, scientists are literally steering the development of life. How cool is that?
All right, Section 4. Step-by-step tissue culture, creating life daily.
So we've talked about the theory of totipotency, and we've got our nutrient recipe. Let's actually head into the lab and see exactly how scientists harness these tools to create life from scratch. And this brilliantly illustrates the whole process. Let's walk through the complete life cycle of a tomato plant in tissue culture. We start with our desired starting material. We call that an explant. You take tiny, sterile pieces of tomato cotyledons, cut them up, and place them on our nutrient medium. Next, you get this regenerating mass of dedifferentiated cells, which is called callus. Thanks to our hormone recipe, that callus develops shoot buds. Those little green shoots are then carefully cut off and transferred to a rooting medium. And finally, that tiny microscopic piece of leaf has fully regenerated into a complete hardened plantlet growing right there in the soil.
Section 5. Incredible real-world applications. Let's talk global agriculture. Okay, let's zoom out from the microscopic petri dish to the macro level. Just think about some of these sci-fi level agricultural solutions that are directly impacting our daily lives right now. Through a process called micropropagation, we can rapidly clone thousands of elite, genetically identical plants in a tiny space, totally independent of the season. Just to put that into perspective, this technology produces over 400 million disease-free bananas annually in India alone. That's massive. And speaking of disease, we can also produce completely virus-free plants. By culturing just the tiny, uninfected apical meristem of a diseased plant, scientists can essentially cure entirely infected clonal crops, like sugarcane and potato.
Now, one of the absolute most fascinating applications here is artificial seeds. Imagine taking a somatic embryo, so an embryo created from a regular vegetative body cell, not from sexual reproduction, and encapsulating it in a protective coating made of a matrix, like calcium alginate. You are basically building a tiny protective cloning pod. It perfectly mimics a conventional seed, complete with built-in nutritional components and growth regulators. This is a total game changer because it allows for the long-term storage and mass propagation of elite, asexually reproducing plant species.
So the crucial point here is that this technology fundamentally breaks the traditional rules of sexual reproduction. Take somatic hybrids, for example. Scientists can use enzymes to just digest away the cell walls of two completely different plant species, creating these naked cells called protoplasts. They then literally fuse those protoplasts together to overcome natural species barriers, combining the best traits from totally distantly related species. Or look at cybrids. This is where we combine the nuclear genome, the main DNA of one parent, with the mitochondrial or plastid genome of another. We are practically building custom cybrid plants by swapping out their cellular engines. It's wild.
Let's move on and see how this builds into the medical field. Because, we aren't just growing food here. Plants naturally produce compounds called secondary metabolites. They use them for defense, but they are incredibly valuable to us as drugs or flavors. Using plant cell cultures, we can turn these cells into literal biofactories. Instead of harvesting huge, sprawling fields of plants, we just grow the cells in liquid suspension cultures to mass-produce high-quality medicines. For example, artemisinin from the Artemisia plant is mass-produced this way as a crucial anti-malarial drug, or capsaicin from chili plants, which is used to treat rheumatic pain. We can generate these continuously, year-round, under perfectly controlled conditions.
Which brings us to a final, pretty provocative thought. We started with a single microscopic cell's superpower to regenerate. And now, we're cloning hundreds of millions of disease-free crops, inventing artificial seeds, combining species that have literally never bred before in nature, and mass-producing vital medicines in laboratory biofactories. The science we've explored in today's Explainer connects directly to the food on your table and the medicines in your cabinet. So if we can already achieve all of this in a simple petri dish, what is the next frontier for plant totipotency? The boundless future of genetic engineering and plant tissue culture is really only just beginning. Thanks for joining me. Keep observing, keep learning, and keep questioning the incredible science shaping our world.